So it's a miserable day in Sydney. You've all just had lunch and it's probably time for a bit of an afternoon nap. So I hope that there's enough interest to keep you awake, but if not, I won't take it personally. So genetics, you probably think, well, I've either got it, I don't, or they wouldn't test me for it because it's I'm not very likely to have a gene fault and therefore it's not an issue for me. Am I doing something wrong? Or is it? Oh, it's construction. I thought it was from there. Anyway, so I'm from the Hereditary Cancer Clinic at Prince of Wales Hospital, and we're very much connected with the Royal Hospital for Women. And so gynecological oncology or women's bits cancers are very much part of the patient profile that we see. Where do I have to point this?
Point it down.
Down? Right. No, wrong one. There we go. So this is the bit where you have a little nap. So, you know, we talk about genetics. That term rolls off the tongue, but what are we really talking about? So the DNA inside our body is a double strand of thousands of genes, and each strand is a mirror image of the other. And I'm sure you've seen the twisted double helix, and each one of those little ladders represents a pair of chemicals. And we've got thousands and thousands and thousands of genes. We've got 2 copies of every gene, one inherited from our mum and one from our dad. The exceptions being those genes that are on the X and Y chromosomes, because us girls have 2 X chromosomes, so we've got 2 copies of all the genes on the X chromosome, but the guys, they've only got 1 X and 1 Y. So the genes that are on the X chromosome, they've only got 1 copy of them, and those a few little unimportant genes that are on the Y chromosome, they don't count for very much. They've only got 1 copy of them, and we don't have any, we don't need them. So each gene is actually a blueprint for a protein that's got a specific function, but for every cell in our body, only some of those proteins are actually needed. If you think about the genes for eye colour, the same genes for eye colour are in our skin cells or in our liver cells or in our heart cells, but we don't express those proteins that turn the iris in our cells blue or brown. They're just those proteins are not turned on in those other tissues. So each gene has a specific function, but it's only actually needed in certain cells. The other thing about genes is that many of them that are, say, involved in a particular function might not be actually turned on, meaning they don't actually make the protein all the time, but only sometimes. Our skin cells are not rapidly dividing. However, if we go home and cut ourselves, suddenly those skin cells are going to start to grow, to heal that wound. And so there are triggers that turn genes on and off, and particularly growth-related genes, things that tell the cell, it's time to divide and multiply, but only in a very controlled way. And then when we're all healed up, the skin stops growing again. Pretty clever, isn't it? Sometimes it gets it wrong, though. Now, the DNA that I spoke of is each gene is made up of 4 specific chemicals, and they're in a pretty precise order. And you may have at some point read about the Human Genome Project, and that now was, at the time, it was absolutely revolutionary and took a huge amount of money and a huge amount of time that we can now probably do for under a few $1,000s in a couple of days. But at the time, it was revolutionary to actually translate the whole genome of a human being. And now there are databases around the world looking at all the things that we share and all the things that there may be some human variation. The things that make us different to each other make up a tiny, tiny part of our genome. And in fact, the things that make us different from other animals is also a tiny, tiny part of our genome. Obviously, the further away we get from a particular living creature, the less we've got in common. But in fact, what we share with most animals is the vast, vast majority of our genes are the same as those in say, most animals. So this is what a simplistic diagram of our DNA looks like. But let's talk about mutations, because people say, oh, I've got the BRCA gene. Well, in fact, that's good, because we've all got 2 BRCA1 genes and 2 BRCA2 genes. As I said, one inherited from our mum and one inherited from our dad. Where it's a problem is if one of those genes has a mutation in it. So we do all talk about people with the BRCA gene, but we should be saying is they have a mutation in one copy of one of their BRCA genes. So what is a mutation? So a mutation is a change in that precise chemical order. It can be that one or more chemicals in that order, that normal order, is missing, or there might be one or more added in, or there might be where there's meant to be chemical A, there's chemical C. So there can be a different mutation put in there. Mutations arise all the time, and Darwin was the one who really put it into a context that if mutations didn't occur spontaneously, we would all be single-cell amoebae. We'd never have evolved into anything. It's only through mutations occurring that organisms have evolved to be different. The giraffe has evolved with a very long neck to accommodate the very tall trees where it was living, for example. So it's only through mutations that an organism changes to adapt to its environment. Now, from a medical perspective, we talk about mutations as really what we mean is pathogenic mutations. That is, meaning when they stop the cell from doing its normal function appropriately, meaning it causes a problem. What we're finding more and more, though, now is variants of uncertain significance. And mostly they are when there's not a group of one or more chemicals missing or added in, but where just one chemical is changed from an A to a C. Everything else is perfect in the sequence up till that point, and everything after that point can be perfectly normal. And that can be a problem because we don't know necessarily what that particular spot is, if that particular spot in the gene is important or not. And if it's not important, then that is of no significance whatsoever. Unfortunately, the more testing we do, the more variants of uncertain significance we find. And now, on average, if we test people for a group of genes, we tend to find variants in about 10% of them. And there are worldwide databases now, and we regularly turn up variants that have never been reported in any other human in the world before. They're probably possibly out there, but they're unified international databases, and we don't know what those variants mean. And that can be one of the downsides of genetic testing, is how do we interpret that information and what does it mean for relatives when we've got a variant and we don't know what it means. We also, as I said, we have these international databases, and they also contain all the known pathogenic mutations as well. So if we're just talking about the BRCA1 and BRCA2 genes, there are literally hundreds and hundreds now of different mutations. Some of them are found in one family only, and others are found in hundreds of families throughout the world carrying the same mutation. And that's particularly common in some communities where that particular mutation arose a very, very long time ago and has been passed down for many, many generations. And there's, say, an example of this is one particular gene fault in the BRCA1 gene, the one that Angelina Jolie carries, that can be traced back to every Jewish population around the world and is thought to have been arose at least 2,000 years ago. And from that, whenever it arose, it's just been passed down from generation to generation ever since, and is now present in that particular gene fault in over 1%. So 1 in 100 people of Jewish ancestry will have that one particular gene fault. Now, if the thing about mutations, if they're pathogenic mutations, it means that a perfect protein will not be made if it's pathogenic. And in many cases it means that the protein will be, a bit of protein will be made, but not all of the protein. And that protein therefore won't link to another protein to do whatever it is the function of that particular set of proteins. Now, mutations that I'm talking about today primarily are the ones that are inherited. They're the mutations that are in every cell of our body and therefore can be present in our eggs or our sperm and can be passed down from generation to generation. But every cell in our body is, throughout our lifetime, has the potential to, when it's dividing, make a new mistake, make a new mutation. And that probably happens scarily much more often than we think. But our cells have their own internal capacity to say, this cell isn't right, and cause the cell to pretty much self-destruct, or its internal repair mechanisms will correct it. But we know that as we get older, more and more of our cells mutate, and we're much more likely to get a problem with a growth-controlling gene as we get older, and that's why we're more likely to get a cancer of some description the older we get. We're also more likely to have mutations in our cells that are susceptible to lifestyle and other environmental exposures. We know what the sun does to our skin, and that's through mutations in our skin causing perhaps a skin cancer, or the mutations that occur in our lungs with exposure to smoking. And all cancers are due to a whole series of new mutations occurring in those cells, in starting off with growth-controlling genes, the ones that regulate if those cells should be dividing or not dividing, how quickly they should be dividing, and also then about those other things that make a cancer cell different. A cancer cell is different not only that it is dividing rapidly and in an uncontrolled fashion, but if you think about it, all our cells need oxygen, and if you've got the same type of cells dividing, then it will need new blood vessels to grow into that cancer to provide it with the tissue, with the oxygen and other nutrients that it needs. And so it has to turn on the genes that cause new blood vessels to grow. It also, we don't break off a breast cell when it's not cancerous and a breast cell suddenly starts growing elsewhere in our body, because cells need to be stuck together. Normal cells have to be stuck together next to other cells of the same type. But cancer cells lose that requirement and are able to travel elsewhere in our body and set up shop somewhere else. And that's all due to different acquired mutations that occur only in the cell. So there's the difference then between the inherited mutations that are passed down from generation to generation and those acquired ones which are just part of the cancer process. There are some cancers though, and this is where it's particularly relevant for some types of ovarian cancer, that are more likely to occur if we're missing a particular faulty gene, or we've got a mutation. And that's where BRCA1 and BRCA2 come into it. So I'm sorry, I haven't been here today to see everything that you've already that you've had discussed, but just to think about, we talk about ovarian cancer. It's not one disease. There are many different kinds of ovarian cancer. And the main one that we talk about when in genetics is epithelial ovarian cancer. Did we say we've got a pointer? Does it?
Yeah, the third one is the pointer.
There we go. It's pretty plain. So this is our women's bits with our vagina here, the cervix. So you can see why a pap smear that takes these cells from here isn't very useful in detecting ovarian cancer, because it's not actually anywhere near our ovaries up here. Our ovaries have their blood supply coming in, and over here we've got millions of stored eggs. All our eggs are made when we're in utero, tiny, early embryos. When the ovaries are developing, that's when all the eggs are made, and they stay there waiting to develop, and one at a time usually, sometimes two or more, but generally only one, are released each month. And then the outer layer of that follicle then produces progesterone, and then that's what keeps everything going till the next cycle goes along. Now, these cells, and then in between them is a whole lot of other cells that hold the whole thing together. Every different type of cell can have a different type of cancer. The cancers from all this internal part, if you think of an orange, the inside part, have very little relationship with inherited genetics. It is only the cells from the outside of the ovary, epithelial ovarian cancer, that is what we speak about when we're talking about genetic cancer. So this is what an ovary looks like. The pathologist will stain the tissue. It's a very, very thin slice. Very thin slice on a glass slide with special stains that show differently in different cells, and this layer, the sort of more orangey layer on the outside, is the epithelium, and that's what is, when we talk about genetic ovarian cancer, we're really only talking about epithelial ovarian cancer, usually with BRCA1 and 2, we're just talking about a particular kind, the non-mucinous kind. Just one other thing that we've discovered in recent years is that the inner part of the fallopian tube that sucks up the egg to take it here for fertilisation and here in the uterus for implantation, that the lining of this part of the fallopian tube is probably where a lot of these serous ovarian cancers actually do occur. They probably don't start from here, but from the same kind of cells that are in this part here. And when we remove the ovaries and fallopian tubes of women, particularly with a BRCA1 gene fault, if we remove them as a preventive, we know that depending on the age of the patient, there may be up to a 5% chance that they already have precancerous changes in these fallopian tubes. And particularly for many women who are diagnosed with cancer, what we call primary peritoneal, where it may be all over the abdominal cavity, it may have arisen in association with a BRCA1 gene fault from the fallopian tube, but we still call it ovarian cancer. So ovarian cancers are not one disease. If we think about the ones that are not from the outside, the internal ones, they're very, very rarely genetic. They're not due to an inherited genetic susceptibility. And there are some very rare kinds with rare disorders, but not BRCA1 and BRCA2. Then if you think about the ones from the epithelium on the outside, they fall into the non-mucinous and the mucinous. The mucinous ones can be due to a genetic susceptibility, but not BRCA1 and BRCA2, but other genes that are involved in a syndrome called Lynch syndrome, which is associated with an inherited susceptibility to bowel cancer. In the non-mucinous kind, the vast majority of them are what we call serous, of which up to about 20% will have an inherited genetic basis. And then there's some other kinds that are less commonly associated with an inherited susceptibility. So meaning that the vast majority of people who get ovarian cancer, it won't be due to any nasty gene they've inherited. It's due to something that's happened along the way in their ovaries and was never predetermined by their inherited factors. It's only a small proportion. 20% is the highest in the serous, 5% in the endometrioid ovarian, and much less commonly in any others. Now, what are the genes? I'm sure you've all heard of BRCA1, BRCA2. They were named because they actually have a higher risk of breast cancer than ovarian cancer. So the breast cancer gene was discovered, and then they quickly realised, well, that only accounts for a small proportion of the families they come across in research, and then they found BRCA2. Then not much happened for, pretty close to 20-odd years before we found any other genes that caused an inherited susceptibility to ovarian cancer. Now we're just starting to test for RAD51C and RAD51D, and we are gathering information about people who inherit a fault in one of these genes. They're probably pretty much just associated with ovarian rather than breast cancer, but we're still getting more information. PALB2, I think I might have some slides in more detail later. I can't recall. I wrote the slides on Thursday. It was a long time ago. PALB2, we don't know if gene faults in PALB2 gene increase the risk of ovarian cancer or not. The interesting thing is these genes all make proteins that work in the same pathway to repair damaged DNA. And but some seem to work more specifically in some tissues than others. So why RAD51C and D don't cause an increased risk of breast cancer, we don't know. But maybe in our breast tissue there's another gene that does the RAD51C and D job if they're not working. And why some genes cause an increased risk of cancer in some tissues and not others is still pretty much a mystery. Oops, did I go? Now, what do all of these genes have in common? So they all repair damaged DNA in ovarian tissue, and this damaged DNA causes the cells to multiply abnormally. The thing is, it's a bit like a car going slowly. If the brakes don't work, it's not as big a problem as if you're driving the car at 100k and the brakes don't work, you're going to do a lot more damage. The same thing is here. Once those cells start dividing quickly, they make more mistakes, and they make more mistakes, and they make more mistakes. So it becomes actually exponential as to the number of internal genetic mistakes in the cell once a cancer starts. And as I said before, when more mutations occur in the cell itself, that enables the cancer to spread. So BRCA1 and 2, what are they? They're actually cancer protection genes. So as I said, if we don't, it isn't technically correct to say someone's got a BRCA1 gene. We all have BRCA1 genes. It means they're good genes. They protect us against breast and ovarian cancer. And for men, they also help protect against prostate and pancreas cancer as well. It's probably around 1 in 250 people that carry a mutated BRCA1 or BRCA2 gene. We always used to say it was about 1 in 500, but there's some newer studies that are suggesting that perhaps there are more people with mutated BRCA1 and 2 genes than we previously thought. even though they work in the same pathway, these two genes they are different if you've got a gene fault. If you've got a mutation in BRCA1, then your lifetime risk of breast cancer is about 70%. And typically those breast cancers are what we call triple negative. That is, they're estrogen receptor negative, progesterone receptor negative, and HER2 negative. People sometimes say to me, estrogen caused my breast cancer, it was ER positive, estrogen receptor positive. It's important for people to recognise that normal breast tissue is ER positive, PR positive. That's what's normal. It's just that those breast cancers that have lost that receptivity are ER negative. It doesn't mean that estrogen caused the cancer. It just means whether or not that cell still interacts with estrogen or not. BRCA1 mutations have about a 40% lifetime risk of developing ovarian cancer, and I've put it in inverted commas there because we also include under that term fallopian tube and primary peritoneal cancers. They're all of the same kind, generally, not all of the same kind, they're typically high-grade serous cancers, and as I said, they may arise in the fallopian tube, so they're not truly necessarily ovarian, but the cells all look the same, behave the same, and respond to the same medications. There is a small increase in risk of pancreatic, prostate and male breast cancer in association with BRCA1 mutations. BRCA2 has a similar risk of breast cancer over a woman's entire lifetime, but the risk is more pushed towards the older ages, and women are not as at such high risk at their much younger ages as they are with BRCA1. And the cancers are pretty much the same spectrum of cancers as we see in anyone else, that is, they're more likely to be estrogen receptor positive. There's probably somewhere around an 18 to 20% chance of developing ovarian cancer if you have a BRCA2 gene fault. What's interesting with BRCA2 is that we see higher risks of prostate and pancreas cancer and male breast cancer than we see with BRCA1. And we also see a spectrum of other cancers popping up in people with a BRCA2 gene fault, which is not, we don't see as much in people who've got a BRCA1 gene fault. Now, it's important to know that these, just to go back to the beginning, these genes are on chromosomes that we all carry 2 copies of, and so there are just as many men with BRCA1 and 2 gene faults as there are women with these gene faults. And half of the women who carry a faulty BRCA1 or 2 gene will have inherited it from their father. And if I had, $1 for every time someone said, I haven't had genetic testing because my mother didn't get cancer and there's no cancers on my mother's side of the family, and completely ignoring what's on their father's side of the family, then I'd have a lot of money, because even amongst healthcare professionals, there is still this belief that if you don't have a family history of breast or ovarian cancer, if your mum was fine, then you couldn't possibly have a BRCA1 or 2 gene fault. And that's not the case, because half of the women will inherit it from their father. So who is more likely to have an inherited mutation in one of these genes? Well, the real key is if anyone has had high-grade serous ovarian cancer, there is at least a 20% chance that they will have a mutation in BRCA1 or BRCA2. And particularly if that cancer has occurred under the age of 70, those women will now routinely be offered BRCA1 and 2 genetic testing, irrespective of their family history. Anyone with fallopian tube cancer, because if we see that it definitely did arise in the fallopian tube, then we're very suspicious it might be due to a BRCA1 gene fault. The things in the family history that will increase the likelihood of someone carrying a being found to carry a BRCA1 or 2 gene fault is if they've got a family history also of ovarian cancer, breast cancer diagnosed under 50, male breast cancer, prostate cancer in a younger man under the age of 70, or pancreas cancer, and particularly if those things occur in combination. I mentioned previously about one particular gene fault that's carried by 1% of people of Jewish ancestry. We know that because that particular population underwent what we call a bottleneck, where they were pushed both geographically to a very small community. The vast majority of the world's Jews in around the sort of 1400s all lived in a small area of Eastern Europe because of the persecutions that they were subjected to elsewhere in the world. And from that gene pool, it would appear to be completely random because there's no benefit that we've been able to discover in carrying these gene faults. But they have found that now about 1 in 40 people descended from that population group will carry one of 3 specific BRCA1 or 2 gene faults. And so if someone has Jewish ancestry, then we will offer them testing for those 3 gene faults. Sorry. So how do we find these gene faults? Well, genetic testing has 2 phases. The first thing is a mutation search, where we do look for multiple genes. It used to be like taking a book from the library where you would just proofread one book at a time. Now it's much more automated, and we can download a lot of genetic material automatically and read multiple sections all at once. And so we're now able to test for multiple genes much more readily. If we do all of that gene searching and we don't find a gene fault, then there's no point offering genetic testing to the unaffected women in the family, to the sisters or daughters, because if the person with the ovarian cancer doesn't have the gene fault, then the people who are healthy are not likely to have it either. However, if we do find a pathogenic mutation, those relatives will be offered their own genetic testing, and that's called predictive testing, because that, if it's for an unaffected person, is predicting whether they are at high risk or not of breast and ovarian cancers. In that case, they will definitely get a positive or a negative result. In the first case, the mutation search, they might get a variant of uncertain significance. And in that case, in general, we don't offer testing to the relatives, the unaffected relatives, for that variant. But if we don't know much about that variant of uncertain significance, and there are, say, 3 or 4 other family members also with cancer, we might see if all of those people all have the same variant, then we'll submit that information to the international curators of the databases, and that might give them enough information to say, actually, we're pretty sure now this variant is causing the cancers in these families and that all have that variant. On the other hand, if we find a variant and we test 3 other people with cancer in the family and they don't have that gene fault, then we can be pretty sure it's a red herring and it's got just part of normal human variation and nothing to do with the cancers in the family. Now, I've been speaking mostly about the mutations that are inherited, that are passed down from generation to generation. But I also mentioned that as part of the cancer process, there are mutations that spontaneously arise in those cancer cells, and that's part of what's driving the cancer along. Now, sometimes those mutations may mean that the cancers are susceptible to specific drugs, and I believe you've already had a talk today about PARP inhibitors. We are now doing a lot more testing of stored tumour samples, particularly on those women who might have passed away and didn't have genetic testing through, for example, the Traceback project. So we will look at now the stored tumour samples, and if no relevant mutation, there are always mutations, but if no relevant mutation to treatment or for the family is found, then there's no further testing. If, however, we do find a pathogenic mutation in the tumour, then if we can access that woman, we will get blood to test for that specific mutation to see if it was a mutation that was just in the cancer and not in every cell of healthy cell in her body as well, or if it was just a mutation just in the cancer cells alone. We can't always access that, so we, in some cases, may need to, if we can get some normal tissue from what's stored in the pathology laboratory, we might try and do it that way. And if we confirm that that patient, that tissue, that mutation was present in the healthy tissue as well as the cancer tissue, then we'll offer the family predictive testing to see if they carry the same gene fault or not. So what are the benefits of genetic testing? It explains why the cancer occurred, and for many people that big why is something that never goes away after they get a diagnosis of cancer, particularly if they're diagnosed at a young age, particularly if they're very young, if they're the only person in the family that has this type of cancer. And so to be able to say to someone, this was in your genetic makeup, you were at high risk of this happening, can answer that question to some extent as to why they developed cancer. As was spoken about earlier today, it might guide treatment in terms of which chemotherapy, newer drugs such as PARP inhibitors, and possibly even trials of newer drugs that are either evolving now